Welcome, viewers, to the Wisconsin Institute of Medical Research, also known as WIMR. This facility is the home of many groundbreaking research labs. In this video, we are going to meet the diligent scientists of the Ricke Lab in the Department of Urology, and they're going to show us some of the fascinating experiments they conduct regularly. Let's head inside. Here we have Adam, a female mouse who is currently pregnant, and we will administer TCDD, also known as dioxin, to her at a non-lethal dose. Dioxin is a persistent environmental contaminant and highly resistant to chemical, biological, and photolytic degradation. It can also persist in subsoil levels up to a century. This is very important. We are not sure what levels of exposure happen with humans on a daily basis. Therefore, we are not certain whether or not fetal exposure to dioxin can be contributing to the onset of diseases later in life. But this study proposes to investigate whether or not fetal exposure to dioxin may be contributing to the onset of lower urinary tract symptoms in men later in life. The dam will give birth to a mixture of male and female pups. However, since for the purpose of this study we are investigating the prostate, a gland that is only present in men, we will select for the male mice out of the group. These male mice will grow to an age of 6 weeks old. It is the equivalent of adulthood in humans. Next, we will perform a surgery on each of the mice. Make an incision along the back through the skin, subcutaneously implanting hormones. E2 is estradiol and T will be testosterone. We then allow the mice to recover for a total of 1 week, making sure that we close their incisions with clamps. The purpose of the subcutaneous hormone implants is to mimic the shift in hormone balance for aging men. We believe that this shift in hormone balance, along with an exposure in the fetal stage to TCDD, may be contributing to the onset of lower urinary tract symptoms in men. To investigate the lower urinary tract dysfunction using mice, we will now look at what a void spot assay is. Each mouse is selected and placed in a cage by itself on filter paper. We provide food but no water to the mice during that time. The mice are allowed to roam freely, and as they do so, naturally they will leave urine spots throughout their cage. About 4 hours later, at the end of the study, we will have a filter paper that looks something like you see on the right. The filter paper absorbs all the urine spots, and now we can use this assay to investigate their voiding patterns. Please stay tuned to the next video to see what that looks like.
Hello everyone, my name is Dr. Deb Garvey, and I'm a postdoctoral researcher here at UW-Madison in the lab of Ricke in the Department of Urology. A little bit about myself and the research that I conduct here in this lab involves looking at lower urinary tract dysfunction using mouse models. So we're investigating the role of a specific receptor, estrogen receptor alpha, and how it may be contributing to the development of lower urinary tract dysfunction in specific tissue types. So I have 2 different transgenic mouse models that I work with, also with a control mouse model, which is the wild type, which has no genetic alteration. And what we do is that we take those mice and we allow them to urinate on filter paper for about 4 hours. We collect that filter paper and we let it dry, and then we are going to image it using our imager and our software to see what those urine spots and the urine behavior of those mice are on the filter paper. So let's get started. I have a full stack here of my urine samples. So I'm going to take each and place one at a time into the imager. I'm going to keep the doors slightly open for the moment. Open up my software. Now I'm going to set up what the protocol is to image my filter paper. So I have to select an application. And then select the type that I'll be using to image the paper. So in this case, it will be rubidium bromide. Then I'm going to select the size of the filter paper. I know that it is 28. 21. So it has the full scope of the paper. Set the exposure time to 0.5 seconds, so it's pretty quick. And now I'm going to position the gel, and also the computer makes me know that I have it on the right filter. I have filter 1, so I say okay. Now it shows me what the filter paper looks like in the imager. It has a nice grid pattern. So now I'm going to open the door and readjust it so it sits perfectly in the center. That's about good. Make sure the door is shut. Now I can run my protocol. It takes only about a few seconds in order to scan the entire filter paper. And then it produces a picture like this. So it is in a grayscale, but now I can see all of my urine spots, and it is way easier to see than looking at the filter paper itself using only my eyes. Now this image I'm going to export for further analysis using different software that I will show you in just a moment. So here you see in front of you on this monitor is an image downloaded from our imager of the filter paper that the mouse had urinated on. So now the image is grayscale, but it is pretty dark. So we want to be able to visualize these urine spots a little bit more clearly. So the first thing we're going to do is adjust the levels for the color of the actual urine spots. This is our white point. We're just going to make the spots a little bit brighter so that we can see exactly where they are. Perfect. Now in this case, we want to be able to also keep all the spots together in terms of circulating them. So now we select this little selection tool and make sure that it is on a circle loop. And we simply click on the spot and draw a little circle around the entire spot. And then from here, we're just going to crop it out. And we do this throughout the entire image. And we continue on with this process until we have selected all of them. Such as in this case is a little bit simpler. We only had 2 urine spots, so we only had to select 2. But now we need to make sure that the background has a full contrast with the actual urine spots. So in this case, we're going to select our levels again, and this time black out the background. And try to get it as dark as possible. Alright, that looks great. Now with this image, we're going to download it into another software. And this software will actually quantify the size of our urine spots, along with their location and how close they are to the periphery, and how many of them actually exist. So let's give this a go. Alright, now that we have our image of the filter paper color corrected so that our urine spots are white with the black background for perfect contrast, we are now going to crop this image for further analysis. So selecting our cropping tool plug-in, and then we're going to draw a line right across the center of the image. Hold down our shift key so that we have a nice straight line. And we click OK to continue. And from here, we're going to select the width. So from the center of the image, how wide do we want it to select? And in this case, we set it at 900 pixels. Then you're going to get a wide bar. It's going to do this for all of the images that we selected that were in the folder, and now you can see what that 900 pixel width looks like now that we selected for it. And it's going to automatically bring up each image, making sure that we have all the spots within that thick bar. Not quite in the middle in this case, so I could just bring it down. And now that we have everything, we click OK. Now we select our next software plugin, and that is the Void Wizard. Now this is really cool because this is where it's going to give us a perfect analysis of all the images that we just set with our pixels. We select a folder that we want it to look in. And now here it's running and it's analyzing each of the images. And now in this case, it did its own recorrection of all the spots. So now they're nice and clean and perfectly quantitated. So for each mouse, this is the label ID, and then we have how many spots in each case. So in terms of the first mouse, we have 7, and then as we look at the other images that had plenty more spots, it gave us 58, 92, and a whole 126. And now these other columns represent the size of the spots and how many fall within that range. This data we then use to summarize and figure out what are the differences between the mice per treatment and also based on their genotype. That is how we analyze our DSAs. Hey Christian.
Oh, hey Dad, how's it going?
Pretty good. I just gave a little bit of what I do here in the lab, and I was wondering if you could tell our viewers about what you do.
Yeah, absolutely. Hi everyone, my name is Christian Ortiz Hernandez. I am a third-year graduate student here at the University of Wisconsin-Madison. Just like Deb, I work in the lab of Dr. William Ricke, and we're in the Department of Urology. Now, I'm originally from the island of Puerto Rico, so minus the cold weather here in Wisconsin, I've really enjoyed my time here, experiencing different, learning different techniques, getting the work done to eventually earn my PhD. So yeah, I thought I'd pretty much show you guys what I was doing, but I guess I'll start talking about what it is that I do here at the University of Wisconsin-Madison. So what I'm generally interested in is lower urinary tract symptoms and how they might develop in Asian men. So lower urinary tract symptoms are a series of urinary complications that can occur, for example, emergency urinating, excessive urinating at night, among other different symptoms, right? But as a field, we still really don't understand fully what are the different mechanisms that can lead to lower urinary tract symptoms. And there are different factors that can contribute to this. One of them might be benign prostatic hyperplasia. So this is a disease in which the prostate in AE men starts getting larger and larger. And that could definitely contribute to urinary complications. However, my project specifically and what we do at the Ricke Lab, we're pretty much interested in other alternative mechanisms that can also lead to urinary complications, such as fibrosis, for example, prostate fibrosis. So fibrosis, in general terms, when you have an increase in collagen protein being deposited in the prostate, this can lead to different sorts of urinary dysfunction or complications. And my project specifically wants to understand at the earlier steps what are the actual causes of prostatic fibrosis that can eventually lead to urinary dysfunction. So you might have heard about different steroid hormones such as testosterone, for example, estrogen specifically, and my project focuses on how estrogens and these hormones can influence or cause prostatic fibrosis. As scientists, and as you know, in order for us to answer the different questions that we have, we need to dig into a different set of tools, right? And one very important tool that scientists here in urology and among other different scientific communities have used are cell cultures. And this exactly was what I was about to do before you came in here. So I thought I'd show the audience and the viewers back home what it is that entitles using cell culture and kind of go from there.
Sounds great.
Okay, so now let's get started with doing our cell culture experiments. Like I mentioned, I'm interested in how steroid hormones might influence prostatic fibrosis. So what I'm essentially going to do is take some cells, grow them in a flask, wait an appropriate amount of time for them to grow happily. In media, of course, that's going to be the nutrients that the cells are going to be using to grow. And once they grow to the appropriate amount that I want them to grow and they're ready for the experiment, I'm going to actually treat them with different doses of 17-beta estradiol, or E2, which is considered a type of estrogen. After that, I'm going to then collect the cells, absorb them, and then look at different changes in protein after treatment. I'm also going to be looking at what genes are also activated after I treat them with the estrogen, and I use them for my project specifically. So right here you can see on the scope, these are the cells that we are going to be plating soon on the biosafety cabinet, and we're going to be growing them for the rest of my experiments. So we're going to then move on to the hood, and I'll show you how we can get this done. So essentially everything that goes from the inside of the hood is considered sterile. So we have to make sure that everything in there maintains or keeps being sterile. If you have contamination, this can really damage your experiment. So every time you go in, or something goes in, or your hands go in, they must be properly disinfected with, if this is sample ethanol, 70%, for example. So essentially we're going to get started. I'm going to be spraying ethanol, making sure it's all good. And once I go into the hood, again, the most important thing for us to remember is making sure to always maintain a sterile field. So now I'm going to be taking the flask that I've already prepared. Like I showed you, they contain already the media that's going to make sure that the cells grow. I have in here all the media that I need. I also need to make sure I wash the cells before I put fresh media to it, and this is what this buffer is for. We also have our vacuum trap going here, which is going to aspirate off the old media that I want to take out. And we also have, we have pipettes, and we also have, for example, this pipetter gun, which again, sure is disinfected. And we're going to go ahead and grab the cells real quick. So you have the cells here. Trying to keep your hands at all times. And we're not going to use these flasks for now, but what we want to do now, so these are the cells that need to be transferred from this older flask into the new flask for me to start the experiment. So what I'm going to do is aspirate off the old media, give them a little wash, and then the cells are going to actually stick to the bottom of a flask, like I mentioned before. So we need to do a little treatment with trypsin to be able to make sure to detach the cells so that I can transfer them to the new flask. So I'm just going to go ahead and do that. So every time you're working with different flasks of cells, and again, to be able to make sure that there's no contamination, you want to make sure the tip of the pipette doesn't touch anything. If you do, it has to be thrown away here. This is just some mixture of water and bleach. So we can get started. We're going to pull in to the bottom, aspirate off the old media, waiting for it to aspirate off completely, and then gently making sure it doesn't spill or anything, put in the pipette there, and you're going to repeat the same thing with the additional flask. put in here, same thing, aspirate off the media. Now I want to be able to rinse the cells again with the buffer that I mentioned. So we're going to take this off and we're going to bring over a pipette. We're going to put it here. Again, same concept, make sure that nothing touches over the actual tips. And then you want to aspirate off certain volume. You want to gently wash the cells. We're going to do the exact same thing with the additional flask. Take it off nice and slow. Take out some liquid, buffer, same thing, and in. This step doesn't require the buffer to be in for a specific amount of time. So you can just kind of move it around, make sure it's covering the entire bottom of the flask accordingly, and then we're going to do the same thing and aspirate off once we can. We repeat the same thing with the second flask. And now at this point, the cells at the bottom of the flask need to be detached, like I mentioned. So we're going to take over the solution here that's going to get the job done. We're going to take a pipetter. And we want to add, at least in this case, I'm adding 1 ml of the trypsin to each of the flasks. Try to make sure that it evenly goes in. Take out the affected tip. Then you do the same thing with the other flask. Now this is a very time-sensitive sort of part of the experiment. Now the cells are going to detach, but you want to make sure that it doesn't stay too long and it starts killing the cells before you actually plate them. So after this, you just put them in the incubator. So there are basically different, make sure that the temperature is correct, make sure that the CO2 levels are correct for the cells to stay alive. But in this particular step, I'm going to put them in for a couple of seconds so they can then move forward and plate them in a new plate. So now that the cells are ready, we want to transfer the old flask, the cells from the old flask to the newer flask, and we need to also use media to do this. So around 2 mils of the media is appropriate, and you're gonna dump it on the cells and keep on aspirating on and off for a little while until you decide to transfer an amount of the cells to the newer flask. Make sure to spread the cells around the flask, and you repeat the same thing with the other. You do the same thing. And these flasks are ready to be thrown out, while the remaining flasks that I'm going to use for my future experiments are going to go back to the incubator.
Hello, my name is Petra Popovic. I'm originally from Hungary, and I came here to study prostate inflammation and fibrosis. And I'm going to talk to you a little bit about tissue processing. So when we obtain the tissues from mice, we fix them in paraformaldehyde. Then we replace the paraformaldehyde to ethanol. And then what we will do is we will embed these tissues. But first of all, we sort all the tissues into these cassettes. Since mouse tissues are very small, we can put more than one tissue into one cassette. But we also have to know what tissue belongs to what mouse. That's why we paint them, and you can see the different colors on this block. This is a paraffin block, and that is basically important to have the tissues embedded in paraffin because we want to get really thin slices, and paraffin is like rocks. It's easy to section and it enables us to have really thin cuts of this block. At the end, we put all these tissues into these slides, and that's when we can start doing the process called immunohistochemistry. Our lab is interested in the urogenital system, specifically in the prostate. The prostate is only found in males. It secretes fluids that nourishes and protects the sperm. However, with age, the prostate often undergoes pathological changes that will also cause urinary symptoms. On the right, you can see the human prostate, which is a walnut-sized gland with different zones. The zone that mostly affects urination is the periurethral transition zone. On the left, you can see the mouse prostate, which consists of 4 different lobes: dorsal, ventral, lateral, and anterior. When we collect the tissues from mice, we collect all of these lobes and process them. When we want to identify where a protein is localized in the tissue and whether its levels change in disease models or in response to treatments, we use a technique called immunohistochemistry. For this, we use antibodies that specifically recognize an antigen that is part of the protein we are interested in. When this so-called primary antibody is bound to the antigen, we add the secondary antibody, which will bind to it. The secondary antibody is conjugated to an enzyme, such as peroxidase. If we add a substrate of this enzyme called DAP, it will produce its conversion from no color to a brown color product. And this is the signal we are going to detect using the microscope. So you can see here that some cells are expressing the protein of interest, which are brown, and the others are not stained.
Hello, my name is Elise Schroeder, and I'm a junior here at UW-Madison, and I am an undergraduate researcher here in the Ricke Urology Lab. And today we're going to be walking through the immunohistochemistry protocol and process. And we have 2 slides prepared here with mouse kidney tissue on them. So the first step in this process is we're going to be baking them at 60 degrees Celsius for an hour. The next step in the protocol after baking and after the slides have reached room temperature is the rehydration process. We begin with xylene, which removes the paraffin off the slides, and then you continue down through the steps through decreasing amounts of concentration of ethanol, and then you end by putting the slides in a tub of tap water. The next step in the protocol is antigen retrieval, and this step is in order to expose the antibody binding sites. So we will use the decloaking chamber, which is essentially a very smart pressure cooker. The next step in the protocol, we will prepare a humidity chamber for our slides. The humidity chamber is so the tissues on the slides do not dry out for the next steps. We will apply a blocking agent and then the primary antibody to our tissues.
everyone, my name is Keegan and I'll be taking over for Elise today and teach you how we do immunohistochemistry on day 2. So I have taken out the slides from the cold room, which have been sitting in there overnight. They still have primary antibody on them, so we're going to wash them a few times with our cleaning solution. This is going to remove any primary antibody that remains on the slides. We will then apply a secondary antibody, which contains an enzyme that will interact with the DAB stain that we will apply. This reaction creates a brown color, which we can analyze in the microscope, which we will see next. Our next step is the DAB staining. I brought the slides over to our small microscope. You will place the slides on the microscope and apply the DAB solution. With the microscope, we will then monitor the reaction progress until we have our desired intensity of stain. We will then apply hematoxylin, which is a nuclear stain, to counterstain our slides, and then go over to the hood, and we will dehydrate our slides, and then cover slip them for further analysis.
This is me again. I would like to show you now the end product of the immunohistochemistry. This is a bright field microscope, and you can see here on the screen the brown staining. In this case, we looked at collagen staining. Collagen is the most abundant protein in the body, and it's important for making up the structure of the organs, the tissues. But what happens in fibrosis is that the collagen levels go up and the tissues become stiffer, so they cannot actually provide the same function as they did before. And here what you can see is a normal looking prostate. Brown stain is around the glands. It's the collagen. But if we go to this tissue, you can see that part of the prostate here has higher levels of collagen, and that indicates that there is a fibrosis, a fibrotic process that's going on in this prostate. So this is how we are analyzing immunohistochemistry, and now Teresa is going to talk to you about fluorescence staining.
Hey everyone, my name is Teresa. I'm a scientist in the lab, and what I'm going to show you right now is how we do some imaging. So you should have already seen how the tissues are stained using something called DAB, and that shows a staining on the slide under white light. What I'm going to show you here is how we can do the same technique, but now use fluorescence. And with fluorescence, we can see many different proteins on the same slide because now we can look at different colors. So with this microscope, we can actually look at up to 9 different colors on one slide. What I'm going to show you is how we do the imaging for that. So what we have here on the microscope right now is just one of the colors that we can visualize under the scope. Right now we're looking at the staining for the nuclei in the cells, and that's what you're seeing here. When you first look at it under the microscope, it will look like a black and white image, and then once we finish taking the picture of all of the different channels, the software has a way to pseudo-color everything. So in general, your nuclei are going to look blue, and then we can decide what other colors the different proteins are in the cell. So with this microscope, what you can do is you can look on the different channels to see different proteins. So this is a prostate tissue, and we can cycle through the different filters to look at different colors. So this is the nucleus, or the blue channel, and then we can change the different channels to look at different fluorescent proteins. So in this channel, we are looking at the green, or what some people call the FITC channel, and so you see the image on the screen looks a little different than what the nuclei looks like. And then we have a red channel, so this one is called Psi3, and so you can see here this protein is less abundant than the other proteins before. And then we have a second red channel called Texas red, and you see again the pattern is slightly different, and then as we focus in and out, you can see the different patterns. And the very last channel that we have on this particular slide is called Psi5. And what's interesting about this is if you don't look at this channel under the microscope, you won't be able to see this wavelength of light by eye alone. So when we stain with this color, you have to look at it through the camera of the microscope, otherwise you wouldn't be able to see it. So these are the channels that we have here, and with this microscope what we can do is we can take an image of all of these channels together, and so what the microscope will do is all the channels that I showed you earlier, it will take an image at each of the channels, at each of the light wavelengths, and then it'll put it together to give us one picture that we can then pull out and look at the protein expression in each of the channels. So now this picture is done. Because these are fluorescent proteins, we don't want to shine light on it for very long, because the fluorescence will start to diminish. So we'll close the shutter and I'll show you what an image looks like that we've already taken. So here you just have the color image of something that we've taken before, and what you can see is that in blue are the nuclei that we saw on the first channel. And then you can see the different colors that are overlaid on each other for the different proteins that are expressed in this tissue. So with software, what we can do is we can tell it the different wavelengths of the light that we're looking at, and it can decide what we are looking at and separate it in such a way that our eyes cannot see. So if you look at all of these wavelengths, these are all of the different wavelengths that are on this slide. So if we choose it, it changes the colors as we see it. And then we can go through now and look specifically at the different wavelengths so that we can understand better how the proteins are expressed in the tissue for any of the diseases that we're studying, looking at treatments, how different drugs or different conditions are affecting the tissues within what we're looking at. So this tool is incredibly powerful because it gives us the option of looking at multiple different proteins that we're interested in one sample, and we can then decide, are the proteins that we're looking at expressed at the same levels in the same cells, or if there's something different. So this tool gives us the opportunity to look at many things at the same time.